Chapter 12, Section 1, The Components and Structure of DNA. You might think that knowing genes were made of DNA would have satisfied scientists, but that was not the case at all. Instead, they wondered how DNA, or any molecule for that matter, could do the three critical things that genes were known to do. First, genes had to carry information from one generation to the next. Second, they had to put that information to work by determining the heritable characteristics of organisms. And third, genes had to be easily copied because all of the cell's genetic information is replicated every time the cell divides. For DNA to do all that, it would have to be a very special molecule indeed. DNA is a long molecule made up of units called nucleotides. As Figure 12.5 shows, each nucleotide is made up of three basic components: a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. There are 4 kinds of nitrogenous bases in DNA. 2 of the nitrogenous bases, adenine and guanine, belong to a group of compounds known as purines. The remaining 2 bases, cytosine and thymine, are known as pyrimidines. Purines have 2 rings in their structure, whereas pyrimidines have 1 ring. The backbone of a DNA chain is formed by sugar and phosphate groups of each nucleotide. The nitrogenous bases stick out sideways from the chain. The nucleotides can join together in any order, meaning that any sequence of bases is possible. If you don't see much in Figure 12-5 that could explain the remarkable properties of the gene, don't be surprised. In the 1940s and early 1950s, the leading biologists in the world thought of DNA as little more than a string of nucleotides. They were baffled. The 4 different nucleotides, like the 26 letters of the alphabet, could be strung together in many different ways. So it was possible that they could carry coded genetic information. However, so could many other molecules, at least in principle. Was there something more to the structure of DNA? Chargaff's rules. One of the puzzling facts about DNA was a curious relationship between its nucleotides. Years earlier, Erwin Chargaff, an American biochemist, had discovered that the percentages of guanine and cytosine bases are almost equal in any sample of DNA. The same thing is true for the other 2 nucleotides, adenine and thymine. The observation that A's are equal to T's and G's are equal to C's became known as Chargaff's rules. Despite the fact that DNA samples from organisms as different as bacteria and humans obeyed this rule, neither Chargaff nor anyone else had the faintest idea why. X-ray evidence. In the early 1950s, a British scientist named Rosalind Franklin began to study DNA. She used a technique called X-ray diffraction to get information about the structure of the DNA molecule. Franklin purified a large amount of DNA and then stretched the DNA fibers in a thin glass tube so that most of the strands were parallel. Then she aimed a powerful X-ray beam at the concentrated DNA samples and recorded the scattering pattern of the X-rays on the film. Franklin's worked hard to make better and better patterns from the DNA until the patterns became clear. The result of her work is the X-ray pattern shown in figure 12-6. By itself, Franklin's X-ray pattern does not reveal the structure of DNA, but it does carry some very important clues. The X-shaped pattern in the photograph in figure 12-6 shows that the strands in DNA are twisted around each other like the coils of a spring, a shape known as a helix. The angle of the X suggests that there are two strands in the structure. Other clues suggest that the nitrogenous bases are near the center of the molecule. The double helix. At the same time that Franklin was continuing her research, Francis Crick, a British physicist, and James Watson, an American biologist, were trying to understand the structure of DNA by building three-dimensional models of the molecule. Their models were made of cardboard and wire. They twisted and stretched the model in various ways, but their best efforts did nothing to explain DNA's properties. Then, early in 1953, Watson was shown a copy of Franklin's remarkable X-ray pattern. The effect was immediate. In his book, The Double Helix, Watson wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race." Using clues from Franklin's pattern, within weeks, Watson and Crick had built a structural model that explained the puzzle of how DNA could carry information and how it could be copied. They published their results in a historic one-page paper in April of 1953. Watson and Crick's model of DNA was a double helix in which two strands were wound around each other. A double helix looks like a twisted ladder or a spiral staircase. When Watson and Crick evaluated their DNA model, they realized that the double helix accounted for many of the features in Franklin's X-ray pattern, but did not explain what forces held the two strands together. They then discovered that hydrogen bonds could form between certain nitrogenous bases and provide just enough force to hold the two strands together. As figure 12-7 shows, hydrogen bonds can form only between certain base pairs, adenine and thymine, and guanine and cytosine. Once they saw this, they realized that this principle called base pairing explained Chargaff's rules. Now there was a reason that A's equal to T's and G's equals to C. For every adenine in a double-stranded DNA molecule, there had to be exactly one thymine molecule. For each cytosine molecule, there was one guanine molecule.